Jane Mwangi. My research journey started back in 2015 when I joined Pwani University to pursue a bachelor's degree in microbiology, and that is where I was introduced to bioinformatics at around 2018, where I was enrolled as a student on attachment here at KEMRI-Wellcome Trust Research Program on bioinformatics. Later on, I interned at PUBREC, that is Pwani University Biosciences Research Center, for 1 year, and then I later got a 2-year master's fellowship in bioinformatics under IDeAL and ENBIT. My master's research work was on genomic and subgenomic characterization of SARS-CoV-2 samples collected in Kenya. Here at KWTRP, we have implemented a genomic surveillance platform for whole genome sequencing of SARS-CoV-2 samples, which implements both Illumina and Oxford nanopore technologies. So the use of this ACTIC protocol has allowed us to sort of quantify these subgenomic RNAs. And the key finding is the expression of ORF10, which on its presence indicated a severe case or deceased case, as was seen in some of our samples, and which has also been reported previously in other studies. However, some other studies were actually contrary to our finding, and which indicated read abundances are quite limited or low number of reads supporting that particular ORF. So this ongoing study utilizes this genomic sequence data to understand the biology of the virus in terms of these subgenomic RNAs. And we're actually keen on finding out more on their role in terms of disease severity and pathogenicity.
My name is Lewis Murugu. I am a former MSc Immunology student at the KEMRI-Wellcome Trust Research Program. I joined the program in 2019, and before that I did my undergraduate studies at the University of Nairobi, and then joined Chapaigo, where I worked as a research assistant. So then I transitioned to KEMRI as a master's student and joined Professor Faith Osia's group, where I've been working on bioengineering of monoclonal antibodies for enhanced immune function. So basically my work was to try and improve function of antibodies specific to the malaria parasite. So what we've seen is that the antibody is important for immunity against malaria, and monoclonal antibodies are coming up as potential new tools for malaria immunotherapy. So for my work, I was interested in trying to improve the function of some of the antibodies that we've expressed and gotten from the group. So to that end, one of the antibodies that I've been working on showed a 2-fold increase in neutrophil activation, and that's an exciting thing, as some of these therapeutics might be evaluated in small animal models for future studies.
My name is Kelvin Mokaya Abuga, an IDeAL PhD student. I joined the program in 2017 as a postgraduate diploma student, and I started the role of the iron regulatory hormone hepcidin in children with severe malaria and antyphoidal salmonella bacteremia. I found that children who had severe malaria anemia had an increased risk of invasive salmonella infection, probably due to disrupted iron regulation. I was curious as to why it was only children with severe anemia rather than other forms of malaria, and I applied for a Wellcome International Master's Fellowship in 2018, which I was successfully awarded, to study the role of iron in children with severe anemia and invasive bacteria infections. As part of the fellowship, I undertook a master's in immunology at the London School of Hygiene and Tropical Medicine. Recently, I was awarded a Wellcome Training Fellowship to further probe into the mechanisms underlying the association between severe anemia and invasive bacteria infections. The basis of my project is on an observation that children with severe anemia have an increased risk of bacteria infections, and I hope to test the hypothesis that disrupted iron regulation and impaired immune function play a role in this association through my PhD work, which started last month.
My name is Marco Tiende. I'm a third-year IDeAL PhD student registered at the London School of Hygiene and Tropical Medicine. Prior to starting my PhD, I was employed as a statistician at the KEMRI-Wellcome Trust Research Program, and my job was primarily to analyze demographic trends and levels in the population that is being surveilled by the KHDSS. So my PhD is generally about understanding the mortality landscape in this population. and varies from trying to describe what the mortality experience is to understanding some of the factors that mitigate and maybe some which are also risk factors for mortality. And a key one, given the current times that we are in, is to look at the impact of COVID-19 on mortality in this population. And we've done some an analysis on excess mortality, and the key findings that we have or we have observed are that for the first 2 or 3 waves of COVID-19, we didn't observe any excess mortality. But between in the 4th wave of COVID-19, which was the Delta wave, sometime between July and September 2021, we did observe a slight increase in excess mortality, but it did not amount to levels that have been observed in other continents.
My name is Kyoko Mikari, and I am a PhD student with IDeAL in the program. Before I joined the program, I was working as a production assistant at a pharmaceutical company in Nairobi. And after compounding intravenous infusions for 15 months, I realized that was not interesting. So in 2016, I came to the program to do my internship. I later went to the University of Cambridge, where I was trained on genomic science at the Wellcome Sanger Institute. In 2019, I came back to the program. I started my PhD, and now I am about to finish. In my work, we explore biological entities called extracellular vesicles that are released by host cells into the environment. They contain RNA, DNA, and proteins, and can be transferred in our blood to remote regions far away from their origin. In my current project, we analyzed the RNA content of vesicles obtained from cerebral malaria patients and found that those vesicles contained RNA from the brain, suggesting that that's where they are originated from.
My name is Mary Bita. I am a 4th-year PhD student registered at the University of Oxford. I joined KEMRI-Wellcome Trust Research Program in 2015 as the first cohort of postgraduate diploma students in health research methods, which is done in collaboration with Kwani University. My research work has been around mental health interventions that aim to address the gap between people with these disorders who access care and those who do not. And I tackle 2 key components. One is access to care, and the second one is stigma, which forms a core component of my PhD work. For the work around stigma, I was funded by the Wellcome Trust Public Engagement Fund in 2019 to set up a community-level awareness campaign, which we dubbed Difusimo. And Difusimo uses participatory approaches, which means that stakeholders in mental health are involved in developing, implementing, and even evaluating interventions to address stigma. And in total, we have about 14 collaborators that work on this project, and preliminary analysis of our work that measures different components of stigma show that these interventions are indeed effective. My future hope is that we can be able to evaluate these interventions using more robust research methods such as randomized control trials, and we can even employ other research methods like qualitative methods to understand exactly how and why these interventions work, with the aim that our work will feed into the wider global work of trying to bridge the gap in mental health.
Makobu Kimani. I am a medical doctor, and my journey began about over a decade ago. I graduated from the University of Nairobi as a medical doctor, then went on to do my obligatory work with the government. I worked in Western Kenya for a couple of years, and then shortly after that I applied and got taken up at the University of Nairobi for a master's in public health. Shortly after that I got very interested in research and started doing HIV-related research in key populations. At the time I was working with female sex workers. In 2016 I applied and was accepted for a Wellcome Trust funded Delta's studentship, a PhD studentship, where I looked, my work was primarily on HIV prevention, again in key populations, but I shifted a little and now I was working with men that have sex with men and transgender women. A key finding of that work was, from my work I produced what was the first HIV incidence estimate in Africa for transgender women. I was awarded a PhD in 2021 by the University of Amsterdam. I have since transitioned into vaccinology on the back of the COVID-19 pandemic. I am now leading COVID-19 vaccine studies. I am currently awaiting the results of a large application for fractional dosing of the AstraZeneca COVID vaccine. Thank you very much.